AAV2 genome map. Understanding this is important. Wild-type AAV contains the rep, replication, and capsid gene. The rep gene codes for 4 proteins and the cap gene codes for 3 proteins. In recombinant AAV, the rep and cap gene are spliced out and removed and supplied in trans. The therapeutic gene of interest is then inserted in the empty space, thereby forming the recombinant therapeutic AAV, RAAV. RAAV also behaves in the cell nucleus just like wild-type AAV. In the absence of a helper virus, RAAV integrates as mentioned above on a specific site on human chromosome 19 via the latency pathway. In presence of a helper virus such as adenovirus, RAAV follows the productive pathway by replicating and forming many small RAAV variant particles. This slide illustrates the underlying technology for small-scale production of recombinant AAV. A mammalian cell line such as HEK293, human embryonic kidney cells, is triply transfected by 3 plasmid vectors. 1. RAAV vector with therapeutic gene. 2. Rep and cap plasmid supplied in trans. 3. Adenovirus helper factor plasmid. Delivery of plasmids into cell can be performed by standard electroporation. In the cell factory, recombinant AAV variants are produced. The cells are lysed. RAAV particles are released. RAAV particles are purified by cesium chloride procedure, and finally this results in purified RAAV ready to be delivered into the human body by intravenous, intraarterial, transdermal injection or into the human eye. This is an expansion of slide number 9. Making modifications to the PAAV/ITR vector plasmid will insert varying therapeutic genes. Example, inserting the ACRP30 therapeutic gene will make the RAAV plasmid suitable for treating insulin-resistant type 2 diabetes. Making modifications to the AAV rep/cap vector will alter RAAV capsid nature and determine cell specificity. Example, constructing a rep2/cap2 plasmid and supplying it in trans is important for treatment in insulin-resistant type 2 diabetes. The type of the rep/cap plasmid supplied in trans during the triple transfection will dictate the nature of the RAAV viral capsid and thereby dictate AAV's ability to target certain cell types, cell specificity. For example, the REP2/cap4 plasmid will make RAAV specific for application in human diseases in the eye, retina. This slide illustrates the modification to be made within the AAV cap gene so that the RAAV capsid is able to evade antibody detection after RAAV is delivered into the human body. If the antibodies were to detect RAAV, they would neutralize it immediately, thereby making the therapy ineffective. After production of RAAV variants by triple transfection, small scale, and using bioreactors, large scale, it is quantified and then delivered into appropriate dosages within the human body for human gene therapy. Now I will discuss the underlying logic for human gene therapy in each condition. Cancer. Quiescence. For lung cancer, in the triple transfection of RAAV production, we use the rep2/cap5 plasmid supplied in trans. This plasmid has genetic elements of the AAV2 genome, chromosome 19 site-specific integration, and AAV5 capsid, lung